Hi, my name is Ashley Mulford. I'm a master's student at Loyola University Chicago, and I will be presenting on Transcriptome-Wide association studies identify novel genes associated with chemotherapy toxicity in human lymphoblastoid cell lines from diverse populations. To start with some background, cancer, which is the second leading cause of death globally, is a disease with a lot of variation from patient to patient. Additionally, a patient's genetic makeup impacts their response to different treatments for cancer. Therefore, developing effective chemotherapy treatments requires consideration of genetic variants that occur in different patients. Lymphoblastoid cell lines constitute one effective model for measuring drug response in an individual. Additionally, LCLs are widely available across ethnic cohorts, making them good for a study like this that focuses on diverse populations. Genome and transcriptome-wide association studies identify relationships between genetic variants and phenotype, including drug response. As the majority of GWAS and TWAS have been conducted on individuals from European ancestries, it is important to focus on other ancestries as they must be represented in order to develop more efficient and personalized chemotherapy treatments. That brings me to the methods for this study. The initial data we started with were collected from LCLs that had been treated with 8 different chemotherapy drugs. The chemotherapy drug concentrations were measured with either AUC, which is the area under the dose response curve, or IC50, which is the half maximal inhibitory concentration, and we had LCLs from 3 different ethnic cohorts: YRI, which is the Yoruba population from Nigeria; ASN, which is a combined cohort of Han Chinese individuals from Beijing and Japanese individuals from Tokyo; and CEU, which is a cohort of individuals with European ancestries from Utah in the United States. We used these data to conduct both GWAS and TWAS in order to find genome-wide significant SNPs and genome-wide significant genes. And we then compared across diverse populations using a combined cohort ALL with all the individuals from these 3 separate cohorts to find significant genes and SNPs across populations. Under GWAS results, you can see the 4 Manhattan plots showing the significant SNPs we identified. In plot A, 2 SNPs on chromosome 4 are shown to be significantly associated with daunorubicin for YRI. In plot B, 3 SNPs on chromosome 9 are shown to be significantly associated with carboplatin for ASN. In plot C, 6 SNPs on chromosome 12 are shown to be significantly associated with etoposide for YRI, and in plot D, 1 SNP on chromosome 3 is shown to be significantly associated with cisplatin for YRI. Additionally, these SNPs all constitute novel associations, as none of them were previously identified and recorded in the GWAS catalog. From the TWAS we conducted, we found 4 genes to have significant predicted expression levels using a threshold of p less than 0.1 after adjusting with Bonferroni. These genes are listed in the table under TWAS results. For the ALL cohort and the drug etoposide, STARD5 was predicted to be significantly expressed by the brain cortex prediction model. For the ALL cohort and the drug capecitabine, USF1 was predicted to be significantly expressed by the liver prediction model, and AHCTF1 was predicted to be significantly expressed by the thyroid prediction model. And for the ASN cohort and the drug carboplatin, FAM173B was predicted to be significantly expressed by the minor salivary gland prediction model. After conducting GWAS and TWAS, we followed up on our results by performing functional experiments for STARD5, as this gene had the most significant predicted expression levels from the TWAS results. The predicted expression plot under functional experiment results shows that as STARD5 predicted expression increases, the concentration of etoposide needed decreases. Therefore, for our functional experiments, we hypothesized that the knockdown of STARD5 expression levels would result in decreased sensitivity to etoposide. We chose the lung cancer cell line A549 for knockdown experiments, as etoposide is often used to treat lung cancer. 2 groups of A549 cell lines were used, one control with SI-SCR, which left STARD5 expression unaffected, and one experimental with SI-STARD5, which reduced STARD5 expression to less than 25% at 0, 72, and 96 hours, as seen in plot B. Both groups were treated with increasing concentrations of etoposide. Then relative viability was measured at 72 hours and 96 hours after treatment. Plots C and D show that at both 72 and 96 hours, the group with knocked down STARD5 expression, seen with the purple line, had a higher level of viability and was less sensitive to etoposide than the group with the normal STARD5 expression, represented by the orange line. This validates our TWAS results that higher expression of STARD5 is correlated with a greater sensitivity to etoposide. To conclude, we found 12 unique SNPs and 4 genes to be significantly associated with drug response. Of those, increased STARD5 predicted expression associated with increased cellular sensitivity to etoposide, which was validated through knockdown experiments demonstrating the same result. Lastly, I would like to acknowledge Dr. Wheeler, who mentored me throughout my time working on this project, Loyola University Chicago's Biology Summer and Mulcahy Fellowship programs, and the Dolan Lab at University of Chicago, which provided the initial data and performed follow-up experiments to our TWAS results. Thank you for listening.